Hello, my name is Ana Rodriguez Quesada, and I work at the University of Malaga in Spain. And I want to share with you this short PowerPoint presentation to explain better what you can find in more detail in our presentation as a poster to this meeting. So let's share the presentation with you. Here we are. Our work is entitled Comparison of Two Different Educational Approaches to the Experimental Teaching of a Luminometric-Based Analytical Method. And it's a work that has been done in collaboration between two departments. One is the biochemistry department in the Faculty of Science, and the other is the experimental science, the education department in the Faculty of Educational Science, both in University of Malaga. Well, we have identified a problem. That was that in spite of the increasing use of luminometry as a technique that is very widespread now in experimental and health science laboratories, our students could not get a good formation on this because there is a shortage of practical laboratory experience to illustrate this technique. For this, we decided to develop a new laboratory experiment for the practical teaching of luminometry in the boundary of the areas of applied biochemistry and analytical chemistry. This experiment is based on the reaction that is catalyzed by luciferase. Luciferase is an enzyme responsible for the light production by fireflies and other organisms, bioluminescent organisms, which emit light for the recognition and attraction of their partner. What is interesting, very interesting in this reaction, is that ATP is a cofactor, and that the light that is produced can be correlated to the ATP concentration. Given that ATP can be found in all living organisms, the concentration of ATP has to be correlated with the presence of microorganisms in water, for example, in tap water. Based on this correlation, we have implemented a new laboratory experiment in two formats, either a short protocol or as a complete problem-based learning experience. In both cases, the driving question was the same, was let's detect if this water sample is has a microbial contamination or not. And it relied on the detection of ATP concentration in those samples of water. In the short protocol, it was a kind of cookbook method where students receive a protocol that had to follow in the laboratory as a kind of recipe that they had to follow in just 1-2 hours of laboratory sessions. And then they got some results and calculated the amount of ATP and presented the results in a final report, and then they had to answer some questions in the final exam. Throughout this process, educational researcher could get some useful data for further analysis. In the other approach, it was the problem-based learning. This was completely different. In this case, students did not receive any protocol. They were asked to split into groups of 4 students each, and they were working as corporations. Every corporation received a letter from the teacher with a problem statement. This was that they had to develop a method to detect contamination of microbial contamination in tap water, and this method should be rapid, reliable, and cheap. With only this information, the students want to search the bibliography, the literature, finding all the possible methods to detect microbial contamination, and they had to select those who gave a response to what was required in the statement letter. This brought them to choose a chemical method based on the ATP measurement, and in this and then they tried to implement them in the laboratory. For this, they had to make some commercial inquiry, prepare the reagents, plan the protocols, and all the laboratory work. For many of them, this was the first time they did this, because they were mostly used to receive a recipe to follow in the lab. After their work in the laboratory, they had to present a final report and also make a presentation in both formats. A presentation for technical listeners and also for non-technical listeners in form of a executive summary. Again, along this process, the educational research got a number of useful data. Both protocols have been applied in 3 subjects of similar profiles within 3 different degrees. The short protocol was implemented in two subjects that were very similar and a kind of applied biochemistry, and in the 3rd year of the degree in biochemistry and in the 4th year of the degree in chemistry. And the problem-based learning format was applied in the advanced instrumental technique 4th year for the degree in biochemistry biotechnology. As for the student results, first indication was the comparison between pre and post assessment tests. This test was composed by 8 short open questions that with technical nature. As you see, we were asking here about luminometry, the luminometer, luciferases, etc. If we see here what is the results obtained in these 3 graphs, you see in orange we see the pre-test results, and in blue the post-test results. We can see here that in all the 3 cases the results were very good. They were better in the case of PBL students, but they were also quite good in the case of short protocols. We can say that the students had learned. As a dot line, you see the orange dot line is the full average for the pretest, and the dotted blue line is the full average for the post-test. Of course, that learning goals, as for the learning goals for the 2 versions of the laboratory experiment, the first is that they are not the same, of course. So we have some learning goals that are shared by both, but other are just specific or are work only in PBL. For example, to elaborate a laboratory protocol, to detail material, reagents and instrumentation, to present results to a diverse audience, to summarize conclusion in an executive report, etc., etc. As you can see in this table, the students' achievement of the learning goals was quite good for both the PBL and the short protocol. But we can see here that in those learning goals who were shared by both formats, results were better in the PBL. But in any case, we could consider that in the short protocol, the results were also quite good. As for the students' feedback, we have a student perception questionnaire for PBL students, and in this Likert questionnaire, students were demonstrated that they were very satisfied with this experience. Here you have some commentaries from PBL students. For example, that they found the methodology innovative with respect to what they had used in other subjects. That, for example, they could understand how a laboratory protocol is developed and to establish a plan better than with other experimental laboratories that they had carried out previously. That this PBL had allowed them to approach situations similar to those that may arise in their professional future, and that even when it had required more work and preparation than other practices carried out throughout the undergraduate students, the activity has been worthwhile because they have learned more. So our conclusions are that the detection of microbial contamination by measuring ATP concentration with luciferase is a useful tool to teach the use of bioluminescence to science undergraduate students, either as a short protocol carried out in a single laboratory session or as a long PBL experience. The students' achievement of their learning goals was very satisfactory. Students who follow the PBL approach could, in addition, adopt a series of additional transversal skills that could be useful in their future professional careers. And between these two extreme examples of the protocol in terms of students' dedication, a whole range of intermediate options could be used by educators to suit their course programming and fulfill the learning objectives to be achieved by their students by means of this laboratory experiment. And well, that's all. Thank you very much for your attention, and keep healthy.